In today's lecture, we'll be studying about carbohydrate metabolism. So, how the carbohydrate get metabolized inside a human body that will be covering up in today's lecture. So, we all know that carbohydrate is a biomolecule and is very essential for survival of any living organism. And it is the main source of energy uh, for any human, or you can say, human organism. So in this topic, we'll be covering up the topic glycolysis. We'll be seeing what is glycolysis and what is all about glycolysis. Just to brief you out the glycolysis. Glycolysis is nothing but the breakdown of glucose. So glyco and lysis. If we break down this word into two parts, that is your glyco and the lysis. The gluco means your glucose molecule and the lysis means the breakdown. Lysis is nothing but the breakdown of glucose molecule. This thing will be studying in carbohydrate metabolism. So we begin our lecture now. So the learning objective of this lecture will be studying about the glycolysis. We'll be seeing the introduction, we'll be understanding what is glycolysis in detail. And we'll be seeing the stages of glycolysis. So what are the stages? We have divided the glycolysis into different stages. Then we'll be seeing when the glucose molecule is breaking down inside the cell. So how the energy is produced, because whenever the glucose is broken, we know that glucose is a source of energy. So how that energy production of take place from the glucose molecule, that we'll be seeing in this lecture. Then oxidation of extra mitochondrial NAD plus and H plus ion will be seen, and the difference between the anaerobic and aerobic glycolysis. So there are like some cells inside the human body where this glycolysis occurs in presence of oxygen as well as in the absence of oxygen. So both will be studying up, and there is a very minute difference between aerobic and anaerobic glycolysis, which is based on the end product. What is the end product? Yes, it is like when cycle get completed. So what is the last product we are getting out of from that particular cycle? Or you can say in this case the glycolysis. So let's start our class. So outcomes will be like will be fully understood about what is glycolysis, the statement of glycolysis, the production of glycolysis, the oxidation of extra mitochondrial and express, and the student and you guys will be able to understand the difference between the aerobic as well as the anaerobic glycolysis. So carbohydrate metabolism. So glycolysis comes under the carbohydrate metabolism. So in carbohydrate metabolism, the major pathways are glycolysis, like the breakdown of glucose into simpler molecule, because we all know that the glucose is six-membered carbon, and here we are converting this six-membered glucose into. So what are the major pathways? Like glycolysis is a major pathway of carbohydrate metabolism. Then after the glycolysis, we'll be studying about the citric acid cycle. What is citric acid cycle? When the glucose is getting break down into simple molecule like lactate and the pyruvate. So the end product will enter the citric acid cycle for the production of more ATP. So ATP is nothing but your energy, so the energy currency of any living organism. Glycolysis and carbohydrate metabolism will be studying about the gluconeogenesis, like how the new glycogen is produced inside the human body. Then the glycogen metabolism is how the glycogen is get metabolized, like the glucogenesis, yeah, glucogenolysis. Glucogenesis is nothing but the formation of glycogen. Glucogenolysis is your breakdown of glycogen into constituted metabolite. Now we'll be focusing on your glycolysis. So the glycolysis, the other word, generally in your examination, the examiner may ask you write about this glycolysis from other way. So the other name for glycolysis is your Abenden Meyerhof pathway. So this is a biochemist who understood this glycolysis. So the other name of this glycolysis or abandoned Markov pathway. So when we're talking about the definition of glycolysis, the glycolysis means the oxidation of glucose to give pyruvate in presence of oxygen or lactate in absence of oxygen. It's a very simple definition. When the glucose is getting oxidized, or you can say there is a breakdown of glucose into a pyruvate. The pyruvate is your end product in your presence of oxygen, that is your anaerobic, and whereas the lactate is the end product in the absence of oxygen, that is your anaerobic. And that's aerobic is the presence of oxygen, and anaerobic is the absence of oxygen. So in presence of oxygen, we'll get pyruvate, and in absence of oxygen, we'll get lactate when the glucose is getting metabolized. The cytoplasm of all tissue cells, but it is of physiological importance in tissue with no mitochondria. Example, your mature RCB, cornea, and the lens. Tissue with few mitochondria like testes, leukocytes, medulla of kidney, retina, skin, and gastrointestinal tract. Then tissue undergo frequent oxygen lack. That's your skeletal muscle, especially during exercise. So the site for this glycolysis generally occur in the cytoplasm of all the cells, like of all the tissues which are made up of the cells. It is generally occur in the cytoplasm, and you can say it occurs in the cells with no mitochondria, like your mature RBCs, cornea, or the lenses, which do not contain any mitochondria. And because we know that mitochondria is a powerhouse of any cell, so if any cells like your RBCs, the cornea, and lens, they lack mitochondria, then how they are going to get the energy? So this is the answer for them, because of this glycolysis, because glycolysis only occur in the cytoplasm. Or you can see that the tissues with very few mitochondria, like your testes, leukocyte, medulla of kidneys, retina, skin, and GIT. Or you can say the tissues which undergoes frequent oxygen, like your skeletal muscle, especially during exercise. And we know that during your vigorous exercise, there is a shortage of oxygen to your skeletal muscle. So glycolysis also occur in your tissues which generally have lack of oxygen during stress. So coming to the steps of glycolysis, they are generally classified into two different steps. If we divide this glycolysis into parts, so there are two different steps. Step one is your energy requirement. We need to give energy so that the glycolysis can occur inside the cell, and there is an energy production. After like the second steps, we will get into the energy out of the cell. Okay, so in step one, which requires the energy, the one molecule of glucose is converted into two molecules of glyceraldehyde 3-phosphate. Okay, so what is saying that we require energy to convert one molecule of glucose into two molecules of glyceraldehyde 3-phosphate. Each step requires two molecules of ATP. So the conversion of this glucose into the glyceraldehyde 3-phosphate, we require two molecules of ATP. To carry out this kind of conversion, we require two molecules of ATP. So it is nothing but energy investment or energy required state. Whereas in your step 2, which is energy production, and finally in this step, we'll be getting the energy out of from the cycle. That is the two molecules of glyceraldehyde 3-phosphate are converted into pyruvate. That is your aerobic glycolysis, or lactate, that is your anaerobic glycolysis. These steps produce ATP molecules. In this step, when the conversion of two molecules of glyceraldehyde 3-phosphate are getting converted into pyruvate or lactate, that time will be getting the ATP from the cycle. So now we'll be seeing the cycle in detail. So how the cycle is all about. The first step is your energy investment, or you can say energy we need to give to carry out this first part of the cycle, that is your energy investment phase. So there are total 5 steps are there, where we need to give energy so that the glucose can convert to the simple molecule. So the rationale or the scientific reason behind the energy investment is that the glucose is a 6 molecule biomolecule. It's a 6 member biomolecule. So we need to break it down. if we want to break it out something, we need to put energy. Like suppose we have a huge stone, we need to put a lot of energy when we are hammering on it to break it down. So that is like, if anything which is very big, we require energy to come it down. It's a size reduction, you can say. We are reducing or you are cutting down the components of any bigger molecule into smaller molecules for so that it can be easily produced, it can be easily processed. We require. So there are total 5 steps where the 6 carbon molecule is getting converted into 3 carbon molecule compound. So that is one first thing is of glucose. We know that the glucose is a 6-membered carbon. You can see on the cursor that it is a cyclic compound. It is a cyclic structure which is given here. Generally, it is a linear one, 6 carbon molecule. So we require ATP and the enzyme which is your hexokinase will convert your glucose into glucose 6 phosphate. There is addition of a phosphate group on the hydroxyl group. Okay. So this is your energy. This step required energy, and this glucose 6 phosphate is converted into fructose 6 phosphate by an isomerase enzyme. That is a phosphogluco isomerase enzyme which convert glucose 6 phosphate into fructose 6 phosphate. This fructose 6 phosphate again required energy, that is your ATP, to get converted into fructose 1,6 biphosphate. So the enzyme required is your phosphofructokinase, and this step is also required energy. So how many energy we have given till now? 2 energy. So we have studied in the previous slide also that energy. So we have 1 molecule of glucose, we require 2 ATP to get converted into fructose 1,6 biphosphate. So this is also a bigger molecule. Now the aldolase enzyme is nothing but these enzymes, lase, wherever the enzyme is coming lase, it means it is breaking down something. So this fructose 6 phosphate fructose 1,6 biphosphate is getting converted into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate, and both of them are isomers of each other. Both of them are 3 molecule or 3 carbon molecules. Okay, so these are the first 5 steps where the energy is invested, where the first energy is getting invested at the conversion of glucose to glucose 6 phosphate, and the fructose 6 phosphate is converted into fructose 1,6 biphosphate. So energy required. So now from the previous we have seen this glyceraldehyde 3 phosphate or dihydroxyacetone phosphate is getting converted into, now it will get converted into 1,3-biphosphoglycerate. The enzyme responsible is triose phosphate dehydrogenase. This is an enzyme which converts these 3 carbon molecules into another molecule, and there is an addition of a phosphate group also. So 1,3-biphosphate means at 1 and 3 position there are 2 phosphate group. So biphosphoglycerate, the molecule is nothing but a glycerate, a simple molecule with 3 carbon atom, and there is an addition of 2 phosphate group at position 1 and position number 3. Then phosphoglycerokinase is getting converted. Now that this enzyme phosphoglycerokinase is left on 1,3-biphosphoglycerate to give 3 phosphoglycerate. So now the energy is coming out from the phosphate group. So we are removing 1 phosphate group and we are converting into 3 phosphoglycerate. So conversion of 1,3-biphosphoglycerate to 3 phosphoglycerate, there is a removal of 1 phosphate group. That phosphate is the group will yield 2 ATP. What will it yield? It will yield 2 ATP. So now this 3 phosphoglycerate, the enzyme is your phosphogluconutase will act on it and it will produce 2 phosphoglycerate. This 2 phosphoglycerate is getting converted into phosphoenol with an enzyme called enolase. Now this enolase will convert this 2 phosphoglycerate into phosphoenol pyruvate. This phosphoenol pyruvate is getting converted into pyruvate with the enzyme pyruvate kinase, and it is also give 2 ATP to us. Okay, so this is your energy payoff phase where the total phase like 4 reactions are there. which is giving all these 4 ATPs to us. We are investing 2 ATP and we are getting 4 ATP out of it. It is not the end. We'll be studying more about the ATP production in our upcoming slides now. So now the energy production. So this table shows the energy produced from one cycle of glycolysis. So one cycle of glycolysis when occur in absence of oxygen, or you can say it is an anaerobic glycolysis. So there is a total production of 2 ATP in case of anaerobic, means there is no supply of oxygen. Okay, so how it is happening? We are giving 4 ATP at substrate level and 2 ATP we are giving at 1,3-diphosphoglycerol and 2 ATP we are giving phosphonyl pyruvate. So energy produced during this one is total 4. If you see the backside, we are giving a 4 ATP we are coming out in your energy period. And whereas in your ATP utilized, we are giving 2 ATPs. If you remember your previous slide, we are giving 2 ATPs here. So 4 minus 2 is nothing but 2 ATP will come out in your anaerobic, means where there is an absence of oxygen. When there is a presence of oxygen, this glycolysis is happening. We are getting 6 or 8 ATP when there is a special condition. Okay. So 4 ATP will be getting removed from the acid, but another 4 or 6 ATP will be coming from the oxidation of 2 NAD plus H in mitochondria. So if you see here, we are getting this NADH. This NADH will also contribute to give ATP when it will enter the mitochondria, because mitochondria will be present in the cell. So totally we will be getting and 2 are we are investing. So if we combine this 4 and 4, 8, 8 minus 2 is 6. So 6 to 8 ATP will be coming out from your anaerobic glycolysis. Then coming to the oxidation, how that anaerobic this NAD plus and H plus ions are giving energy. So the cytoplasmic, we know that this glycolysis is occurring in the cytoplasm, and this intermediate NAD, H and H plus they are coming out. This is nothing but nicotinamide, adenine, dinucleotide. This NADH cannot connect mitochondria. However, it can be used to produce energy produces ATP by respiratory chain phosphorylation. This can be done by a special carrier for hydrogen of H plus ion. These carriers are either dihydroxyacetone phosphate or oxaloacetate. So, it requires a special carrier to get inside the production of. This is a shuttle system. We have this NAD plus will transfer that to glucophosphate shuttle. It is important in certain muscles and the nerve cells. The final energy produced is 4 ATP. The mechanism of coenzyme of cytoplasmic phosphate dehydrogenase is NAD plus, and the coenzymes of mitochondria with phosphate dehydrogenase is FAD. The oxidation of FADH is a respiratory chain with 2 ATP. As glycolysis gives 2 cytoplasmic NAD plus H plus ion, so 2 mitochondrial FAD. So this and another shuttle system is your maillard aspirate shuttle. So now we'll be studying about the differences between the aerobic and the anaerobic glycolysis. So in the aerobic glycolysis, if you see there is a two columns we have made, aerobic and anaerobic. So in your aerobic, the end product from the glycolysis is pyruvate, and for anaerobic, your lactate. So means when the glycolysis is occurring in presence of oxygen, we'll be getting the end product, the pyruvate, and when it is occurring in the absence of oxygen, the end product will be lactate. So the energy is coming out as it's obviously in the presence of oxygen. The more ATP will come out from the aerobic, and less energy will come out from the anaerobic. So regeneration of NAD plus through acidic change in the mitochondria and through lactate formation. And availability to the tricyclic cycle in the SCA, the available and 2-pyruvate can oxidize to be ATP and not available as lactate is cytoplasmic substrate. So what will happen if aerobic glycolysis is occurring? So we'll be coming up with 2-pyruvate, and this 2-pyruvate will go into the Krebs cycle of the PCA cycle, where it will get give 30 ATP. 30 plus 6 will probably 36 or 38 will come out from one molecule of glucose. So it will go complete oxidation. And the end product for PCA cycle is nothing but carbon dioxide. But whereas for anaerobic, the lactate cannot go inside the PCA cycle because the pyruvate is the starting material which is required for the PCA cycle. So importance of lactate production in anaerobic glycolysis is that in absence of oxygen, lactate is the end product of glycolysis. The glycolysis will become the glucose getting converted to pyruvate and the pyruvate is getting converted to lactate because it is the end product of glucose when it is getting oxidized in absence of oxygen. In absence of oxygen, NAD plus is not oxidized by the receptor. Conversion of pyruvate to lactate is a mechanism for the regeneration of NAD+. This helps continuity of the glycolysis as the generated NAD will be used once more for the oxidation of another glucose. Then substrate level phosphorylation. This means phosphorylation of ADP to ATP at the reaction itself. In glycolysis, there are two examples: 1,3-biphosphoglycerate plus ADP 3-phosphoglycerate plus ATP. And it is getting what we are getting is phosphoenol pyruvate and ADP enol pyruvate and ATP. The special feature of glycolysis in RBC. So mature RBC contain no mitochondria, thus they depend only upon glycolysis for energy production. Lactate is always the end product. The glucose uptake by the RBC is independent of insulin hormone. The reduction of net hemoglobin, the glycolysis produced NADH plus NH plus, which used for reduction of net hemoglobin. Now we'll be seeing about some of the MCQs what we have studied till now. Based on that, we'll be answering this one. So in a nutshell, I just want to brief out what we have studied till now. We have studied about the glycolysis. Okay. So what is glycolysis? It's a breakdown of glucose to a simple molecule, and the glycolysis is occurring in the presence of oxygen. It is called the end product is pyruvate, and it is the glycolysis is occurring in the absence of oxygen. The end product is your lactate. So there is a total production of 6 to 8 ATP in your aerobic and 2 ATP is produced in your anaerobic glycolysis. And the glycolysis occurs where it will occur in your cytoplasm of the cell. So coming to the MCQs, which of the following enzyme catalyzes the first step of glycolysis? So we all know that the hexokinase, the answer is your hexokinase because it's the first step when the glucose is getting converted into a glucose 6-phosphate. So this hexokinase is required, and this is the energy requirement and energy dependent step. So hexokinase requires the ATP to convert glucose to glucose 6-phosphate. So this is the first step of glycolysis. The answer is your A. And pyruvate kinase is your later coming enzyme, the glucokinase and the phosphatase 1 enzyme, they are all later is coming. So first enzymes, or you can say the first step of glycolysis is catalyzed by hexokinase, which is a general term used for the aerobic degradation of glucose to obtain energy. Anaerobic means nothing but absence of oxygen. The absence of oxygen, if the glucose is getting converted, it means it is a fermentation. The answer is your fermentation. Whenever the cell ATP supply is depleted, which of the following enzyme activity is increased? Whenever the cell ATP supply is depleted, and the energy in the cell is getting depleted, so which of the following enzyme is activity is increased? That is your phosphofructokinase 1 enzyme will activate it. The cleavage of fructose 1,6-biphosphate one aldose and one ketose. We have seen from that side. So this is three molecule carbon. This is one aldose will come, that is a coming which contain an aldehyde, and another will be your ketose which contain a ketone group. So this is your 4 MCQ we have seen. We'll be seeing furthermore MCQs. Dihydroxyacetone phosphate is rapidly and reversibly converted into dihydroxyacetone phosphate. We have seen from the graph, from the table, the answer is your glyceraldehyde 3-phosphate. So the right answer is your glyceraldehyde 3-phosphate. Fructose is 1-phosphate and fructose 1,6-biphosphate and fructose is phosphate, and this they are the wrong answers. And which substrate is used in the last step of glycolysis? So the answer is, we know that is a phosphoryl pyruvate from which we get the pyruvate. So the last step, the substrate which is used in the last step is a phosphoryl pyruvate, which is getting converted into pyruvate. So the 8th MCQ, the high concentration of glucose 6-phosphate is inhibitory to exokinase, and the high concentration of glucose 6-phosphate is there in the cytoplasm, and the glucose 6-phosphate is not functional. The product formed in the last in the first substrate level phosphorylation is glycolysis. The answer is 3-phosphoglycerate. The final is your glycolysis convert glucose into pyruvate. It's not glucose into phosphoryl pyruvate. It's not fructose into pyruvate or fructose into phosphate. Glycolysis is the conversion of glucose into pyruvate or lactate, depend on the oxygen condition. So answer is your A. That is all about your references which can be used to study more about the glycolysis. So just to summarize what is glycolysis, the glycolysis is a chain where there's a conversion of glucose into fructose or lactose, and special thing that if the oxygen is present, it is called aerobic glycolysis in the end product of pyruvate, and whereas the oxygen is absent, the glucose is getting converted into lactate. And the total energy production from aerobic is of 6 to 8 ATP, and for your anaerobic it is of 4 to 8 ATP. But the most important thing is that in aerobic glycolysis, the NADH which is coming out from the glycolysis it will enter the TCA cycle, or you can say it will enter the mitochondrial cycle, and will give further ATP. Whereas and the continuation is that the pyruvate will enter the TCA cycle, and there is a production of 30 ATPs, and the end product for the TCA cycle is nothing but the carbon dioxide. So totally from aerobic glycolysis will be getting around 30 plus 6, so it's 36 to 38 ATP will be getting from one molecule of one molecule of glucose. When the one molecule of glucose is entering the glycolysis and the TCA cycle, there is a total production of 36 ATPs are there. That is all about your glycolysis, and we have studied about this lecture, and in your next lecture we'll be studying about other biochemical cycles. Thank you. Thank you guys for listening to me. Next class. Okay. Thank you.